But hey, okay. Welcome to Science Saturdays, our third edition. I'm your host, Jilly Forrester, and I'm going to be shortly joined by my two guest speakers, Dr. Emma Meaburn and Dr. Katherine Thompson from Birkbeck, University of London School of Science. But before I tell you about our topic today, I want to tell you a little bit about Science Saturdays. Science Saturdays is a series of free short talks, and they're aimed at sharing science with the public. We're in collaboration with the Me Human Project and the National Saturday Club. And the Me Human Project, if you haven't heard about us before, is a public engagement project which is focused around sharing science with the public to better understand our place in the natural world. And the National Saturday Club is a charitable organization making higher education accessible to youths across our nation. So we're here every Saturday morning this month, covering a new topic with two new scientists from Birkbeck. So our topics have ranged from evolution to development, and from life in this universe to viruses versus antibodies, just to showcase the breadth of science of some of the pretty incredible research going on in our School of Science at Birkbeck. And you might be wondering, is this really different from other podcasts or science programs that you've seen in the past? Well, a little bit, I hope, because we really want to make this science accessible to everyone, regardless of your age or background in studying science or interest in science. And while we have a primary objective to make science easy to understand and relatable and certainly enjoyable, we also want to find out a little bit more about our scientists. So for example, we want to understand how our scientists found their way into their current positions. And not only that, we want to understand, is science really just about keenness to detail and equations and chemistry and biology and physics as we learned them in our A levels or earlier? Or can we integrate creativity? Does imagination play a role in the way that we do science? And is it important? Is it an important factor? So we're going to be interrogating our scientists about these two questions. Don't forget, you can join in the discussion as well. This is a live feed going through Birkbeck's University of London's YouTube feed. So you can type into the chat bar, and if you ask us some questions, I'll pick those up and we'll ask those to our scientists during the panel. We'll have time for a couple of questions after each speaker as well. So don't hesitate to ask questions. And just remember, we're here every Saturday morning during May. So you might be having a long lion or a cup of coffee, or you might be just hanging out with the kids or the neighbors. This is science for everybody. So hopefully you can all enjoy it together. So last week we brought you two talks, and they were about viruses and antibodies and how cells transport information between each other. And today we're talking about nature nurture. So this is a slightly different topic, and now it's time for me to introduce our two guests, Katherine Thompson and Emma Meaburn. Hello.
Hi. Hi.
Hi, Katherine. Hi, Emma. So Emma, you're going to first talk to us about the nature side of things. You're going to tell us a little bit about what makes you and then thinking about our DNA. And then Katherine, you're going to tell us a bit more about the nature side of things. So even though you might be born with everything in a certain quality, with certain qualities and characteristics, how they interact with the environment after that point can have huge influence over how our health, for example. And you're going to tell us about pollutants in the air and how they impact our lungs. So we're really excited to hear from both of you today. We're going to start off with Emma. Emma, are you happy to share your slides with us?
Yeah, I'm happy to share. Hold on, it's now saying, it won't let me share. Hold on.
Let's check and make sure you're...
Got it. It's all good. Yeah, we're here.
Okay, Emma, I'm going to hand it over to you. I'm going to mute my video and my audio, and then we're going to come back to you in about 20 minutes and ask you some questions.
Brilliant. Thank you. Thanks, Julie. So I'm Dr. Emma Meaburn. I'm located in the Department of Psychological Sciences at Birkbeck. So she says, let's try and get my slides right. So the focus of my talk is what makes you, you. And this is a question that's occupied my mind as a teenager. So I was adopted as a baby, but I remained in contact with my birth father as I grew up. And I was similar to him in lots of ways, despite the fact I spent very little time with him. And the similarities weren't just physical. I thought we were alike in terms of our personalities, and we were alike academically. And I had an inkling that our shared DNA had to be important. But my evidence was anecdotal. I had a sample size of 1, me, and I was a teenager. What did I know? And actually, this is really, this question about the influence of genes is the focus of behaviour genetic research. So the importance of genetic factors in how alike people are is a question that behaviour genetic research tackles, which is my area of research at Birkbeck. What are the genetic, the nature and environmental factors that contribute to the differences we see between people, or the similarities, and how do they work? So let's first consider what makes us human. So we're built of cells, trillions of them, and contained in the nucleus of every one of our cells is a set of genetic instructions that contains enough information to build from scratch a highly complex organism that's characteristically human. We walk upright, we use language to communicate. And for any individual, these genetic instructions are the same, for the most part, small caveats, but they're the same in every cell of our body. And in fact, we have 2 copies of this set of genetic instructions in every nucleated cell in our body. 1 copy we've inherited from our mother, and 1 copy we've inherited from our father. So this is my drawing attempting to explain this. So here's a cell in the nucleus of our cell. I've just shown a stretch of this genetic information that's inherited from the mother and the stretch that's inherited from the father. So these 2 copies makes us diploid. And this set of genetic instructions are made of a chemical called deoxyribonucleic acid, or DNA for short. And DNA contains 4 basic building blocks or bases. We have adenine, represented as an A, guanine, represented as a G, thymine, represented as a T, and cytosine, represented as a C. And the order, or the DNA sequence, of these bases is very important. And that's because the information contained in this sequence of A's, G's, C's, and T's is what guides biological processes and is needed for cells to function. The information in DNA is used to make proteins and products such as hormones and enzymes that carry out the chemical reactions and processes that are needed for us to grow and are needed to sustain life. When we get different cell types, so I've shown here my brain to represent, we'll have different types of neuronal cells. We also need heart cells, skin cells, liver cells. And we get different cell types due to how and when the information contained in the DNA is accessed and used. So we each have a set of genetic instructions, a genetic code that we've inherited from our parents, a complete copy from our mother, a complete copy from our father. And this is a really interesting bit. So if you only take home 1 point from today's talk, this interesting point is that whilst we all share these set of make a human genetic instructions in our cells, there are differences in the DNA sequence between people in a population. And again, I've tried to illustrate this here. So I have 3 individuals, and in this first base, it doesn't change. It's always an A for everyone. The second base is always a G. When we get to this third base, there are different possibilities. And I've shown in this example that this position here can be a T or it can be a G. And because we inherit 2 copies, and there are 2 possible bases that could be there, a G or a T in this example, there are 3 possible combinations that any individual can have at this position. They could have 2 copies of the same base. This individual is a T, whereas this individual is a GG, or you could have 1 of each. This individual is a G and a T. And if you were to pick any 2 unrelated individuals at random from the street and examine their DNA sequence, you'll find that they differ at roughly 0.5% of the DNA bases. And given that the fact that the human genome is 3.2 billion bases long, that represents a substantial amount of genetic differences between us, up to kind of 19 million different bases. And changes in DNA sequence that occur in germ cells, that occur in our sperm or egg cells, can be inherited. So they pass from the parent to the child. And if this change in DNA code happened many generations ago and isn't too deleterious to health, then it can spread in the population and become common. And you'll see this new or alternate DNA sequence in at least 1 out of every 100 people. In contrast, if the change happened in more recent history, then it can be rare. It can be unique to you or perhaps unique to your family. But importantly, these differences between people are scattered throughout the human genome. They're scattered throughout the 3.2 billion bases of the genetic code. They don't all reside in one place. They're in genes, they're in the spaces in between genes, they're throughout. And the question that behavior geneticists such as myself are really interested in is how important are these DNA differences, these differences in DNA sequence between individuals? How important are they in accounting for the differences between us in terms of how we think, feel, and act? And sometimes there's a really clear relationship between DNA sequence variation and human health. And typically, when we see a very clear relationship between just one DNA change and human health, it's because there's been a change in DNA sequence that's so severe it disrupts a gene in such a way that the cell can't perform its normal functions. And so an example I've shown here, which is quite a common example used, is shown for Huntington's disease. So Huntington's disease is caused by a repeat in the DNA sequence in chromosome 4, and it results in a defective protein. It causes the Huntington protein to become abnormally long, and over time, parts of the brain will stop working properly. So Huntington's is rare and it is severe. If you have this change in the Huntington gene, you will develop Huntington's, and you only need 1 copy of that change. So the father in this family has a single change in the Huntington's gene here. That's necessary and sufficient. He will go on to develop Huntington's disease. And that means that his children, each of his children, will have a 50% chance of inheriting this change. So this child here has inherited the Huntington chain, the change in DNA sequence in the Huntington gene from the father and a normal copy from the mother. This child here won't go on to develop Huntington's disease because they've inherited the unaffected copy from the father and an unaffected copy from the mother. So here, for these rare and severe genetic diseases, there's a clear one-to-one relationship between DNA change and disease. The change in DNA sequence is necessary and sufficient to cause the disease. So these children will go on to develop Huntington's. So a key question then is how important are DNA variants or these genetic differences in explaining differences in how we think, feel and act? Huntington's quite an extreme example. It's rare and it's very, very severe. But what about thinking about mental health disorders? How important are these genetic variations that are common in the population? How important are these common genetic differences in explaining differences in how we think? What about explaining differences in why some people go on to develop depression and some don't? Why some individuals go on to develop schizophrenia and others don't? What about differences in how well we do at school or how clever we are? Are these genetic differences still important? And when we look at psychological characteristics and disorders, we do not see this clear inheritance pattern in families like we see with single gene disorders. There is no clear one-to-one relationship between a change in DNA sequence and a human characteristic or health outcome. So are genes still important? Actually, we can ask if they're still important by comparing the similarity between pairs of related individuals. If genetic differences or genetic variants contribute, then we can predict that the resemblance or similarity between pairs of individuals will increase with increasing genetic relatedness. So, for example, first-degree relatives, such as siblings, will be more similar on a particular characteristic than second-degree relatives, such as nieces and nephews, if these genetic differences are playing a role. And this is exactly what several decades of behaviour genetic research has done, typically by using the twin design and exploiting the fact that identical twins share 100% of their DNA and non-identical twins share on average 50% of their DNA variants. So this is, I think, my only graph in my talk, but I wanted to show it to you because it's from a very kind of famous study that was published a few years ago now. But this study looked at nearly all published twin studies of human characteristics and disorders. And in this study, they wanted to see overall across all these studies, what are the origins of differences between people? To what extent do genetic differences explain the differences we see between people? And that's indicated by the height of these purple bars. So the height indicates the magnitude of genetic influence for the characteristic examined. It can range from 1, that would mean genetic differences are explaining all the differences between people, and 0, genetic differences aren't having any impact. And so hopefully you can see, and to answer the question I always get asked by my non-geneticist friend, which is, what's really in our genes? The answer is pretty much everything. Whether we look at how we think about how we behave, or think about whether or not we go on to develop mental health disorders, genes are important. These genetic differences are playing a role. But also note that these bars, whilst they're present, they're not at 1. Genes aren't the whole story. So DNA sequence differences are important in explaining some of the differences between us, but they're clearly not the whole story. And on average, they're explaining about 50% of the variation in any given characteristic. They matter, but they're not the whole story. So the next question, which is the question I'm really interested in because my background is in molecular genetics, is that, okay, we know that genetic differences exist between people and we know where these differences reside in the genetic code. And we know that they're explaining part of the reason why we're different. But a key question then is, which of these DNA variants are important? Is it this one? Is it this one? If it is important, how are they working? How are they impacting how the information in DNA is accessed and used? Are they changing the function of the cell, and if so, how? And the way we can actually answer this question is to perform DNA studies. So study the DNA directly. And again, basically perform molecular genetic studies, which is what my training and real interest is in. So in order to identify the specific DNA variants involved, their number, their location, and how they work, we need to perform DNA studies. And specifically what we do is we perform something called DNA association studies, where we test for a statistical link between any given genetic variant and the human characteristic or outcome of interest. And I've tried to illustrate this again here with my, my iPad drawings. So in a DNA association study, what we first need to do is to measure and record the characteristic of interest. And in psychology and psychiatry, we've been very good at measuring differences between people on the behavioural and psychological level for a long time. So we might be interested in perhaps looking at brain structure and function, so performing MRI studies between individuals, maybe performing EEG studies when we're looking at electrical activity in the brain and how that differs between people. Perhaps we'll perform psychometric testing, thinking about how individuals think, or use clinical reports or observation. But we're very good at measuring differences, characteristics between people, how they differ. We then, for this is my research participant, for this same research participant, we need to collect DNA. So we collect a DNA sample, and typically I collect DNA from using saliva or from a blood sample. And then we use technology in the lab, such as SNP genotyping microarrays or next generation sequencing technology, to determine what genetic variants any individual has. So we'll say, for this individual, I know that this position changes between people, but what change does this person have? I know this position changes. What basis does this person have? An important point to make is in the last 10 years, and it's only really quite recently, so the last 10 years, we've become very, very good at being able to rapidly and accurately measure genetic variation in large numbers of people. We can measure over 1 million genetic differences in an individual in samples of, in very large sample sizes of upwards of 5,000 individuals, rapidly and accurately. And then we then use statistical approaches to identify which one of those million, possibly millions of genetic variants tested, which one of those DNA sequence variants relates to our characteristic of interest. And because of the huge numbers of tests involved, we look at perhaps 1 million genetic variants in 10,000 individuals. We use high performance computers to able to perform the calculations and to visualize the data and make sense of the results. And I want to give an example of what that looks like when we're doing an association study, what we're really looking at. I want to give an example for any one genetic variant. So this is just an example looking at this one change here. So we know this position, this third position changes or differs between people, and it can be a T or it can be a G. So this individual has 2 T's, this individual has a G and a T, and this individual has a G and a G. When we're performing tests of genetic association, what we're really doing is we're looking at a difference in the frequencies of these 2 bases between our case and our control groups, between our individuals with and without a disorder. So again, I've tried to illustrate this here. This is a very simplified version. Obviously, in reality, this is on a larger scale. But if this genetic variant, if this base change here is involved in risk for a disorder, then we'd expect to see a difference in the frequencies of G's and T's between people with the disorder, the case group, versus those without. And hopefully you can see that I've illustrated that here. So I've got 10 individuals in my group, and then we can count up the number of T alleles. There's 15, and there are 5 Gs. So proportionally, 75% of my case group has a T allele, 25% has a G. When I compare it to my control group, there's a roughly equal number of T's and G's. It doesn't seem to differ. What's important then is these frequencies or these ratios between the case and control groups are different. The T allele is more frequent in the affected. And if this difference that we see is statistically significant, then we can infer that this genetic variant plays a role in the disorder. And specifically, we can say that the presence of a T allele actually increases risk. And we do this in these association studies, we do this for all the common genetic variants throughout the human genome. We call them genome-wide association studies. So we look at upwards of 1 million genetic variants across the genome in large numbers of individuals. And for each one, we ask this question. So this is my example I've just shown. This example here, again, it's a genetic variant, it differs, but there is no difference in the frequencies between case and control groups. So whilst there is a genetic variant here in the population, it's not associated with disorder, whereas this one is. And genetic association studies such as these, like I said, have been around for about 10 years now, and they have massively expanded our knowledge of DNA sequence variants that contribute to psychological traits and disorders. We've identified hundreds and thousands of specific DNA variants that explain a statistically important amount of why people differ in how they think and how they act. The key insight, however, is that there are hundreds of these genetic variants. So there never is and there never will be a single gene or a single genetic variant for human psychological traits and disorders. In fact, the findings are consistent with a polygenic model of inheritance. There are many common genetic variants or DNA differences spread throughout the human genetic code, and together they're contributing to the differences in how people think, feel, and act. And critically, most of these genetic variants on their own have a very, very small impact. So each DNA variant explains a small amount of differences between people, and there are lots of them spread throughout the genome. They're in genes, they're outside of genes, and the spaces in between genes. So the key finding is that there's a very large number of genetic variants, and that each of these variants is inherited in a Mendelian fashion, and they're together contributing to the differences between us. And further, it turns out that even though each of these individual DNA variants has very little impact on outcome, we can add them together and they can sum together into a single genetic score that then explains a useful amount of the differences we can observe or measure between people. And these are called polygenic scores. And so to calculate a polygenic score for a particular disorder or characteristic, what you do is tot up the number of risk increasing or risk decreasing bases or variants across an individual's genome, along with their magnitude of impact. So here, for example, I'm looking at the same basis, but now I'm going to calculate my genetic score for 2 individuals. So we know this change differs between people, and we know from our association study that the T allele, the T base, increases risk for depression. This individual has a T, so let's give them a score of 1. They have 1 T. This individual has no T's. Give them a score of 0. We'll look at the second variant. Whilst it varies, it's not associated with depression, so we'll skip it, we'll ignore it. Let's look at this last one here. Again, it differs. It can be an A or a G, and our association study has told us that the G variant, the G base, increases risk. This individual has 2 Gs, so we're going to give them a score of 2. This individual has 1, we'll give them a score of 1. And these polygenic score means you add up across the genome these scores of increasing or could be decreasing variants to get an overall genetic score. And in both instances here, you can see that my 2 individuals get a score of 3. But note that that score of 3 is coming from different regions across the genome. So they end up with the same score, but that score is actually, the signal from that score is coming from slightly different genetic variants. And also you'll note, I hope, that the maximum score in this very simple example could be 6. Any individual could have both risk alleles, each of the 3 genetic variants. They could have a maximum of 6, or they could have none. They could have a maximum of 0. So in this instance, both of my individuals are kind of scoring in the middle in terms of their overall genetic liability for depression. And again, this is a very simplified example. We do this for hundreds of thousands of genetic variants across the genome. And these genetic scores are interesting because what they're going to try and do is capture an overall genetic propensity for any individual in terms of their liability for a disorder. So in my example, both my individuals had the same score, 3. But it turns out when we calculate these genetic scores in large populations of individuals, we see a range of these genetic scores. Some people will have a higher score relative to everyone else. Some people will have a lower score relative to everyone else. And there's a lot of interest in using these genetic scores to identify individuals at higher or lower risk for psychological traits and disorders. And that would make sense. There's the rationale here is that health and quality of life might be improved if you can identify people earlier who might be at risk. So for example, if you've got identify people with higher genetic scores, perhaps you might want to screen them earlier, or perhaps if they're low, maybe you'll screen them later. Perhaps you'll think about different treatment options or provide treatment earlier, or intervention strategies. Or perhaps this knowledge might empower individuals to make lifestyle choices. However, always caveated the joy of science, it's worth noting that most of us are going to fall in the middle. Like my previous examples, my individuals had a score of 3. Most of us are going to fall in the middle in terms of our genetic scores for most things. We're going to have an approximately even number of risk increasing and risk decreasing alleles. And so it's unclear how these genetic scores might be of value to us. And finally, I have to emphasize, and emphasize this quite strongly, that genetic scores in the field of psychology and psychiatry are nowhere near good enough for prediction. Statistically, we can see a difference in genetic scores between case and control groups, but they're nowhere near good enough for personal use or prediction. So just thinking about that, that means that we'll see individuals with a high genetic score for a particular disorder who won't go on to develop that disorder. And conversely, we'll see individuals with very, very low genetic scores who will. And perhaps that's for other reasons. Perhaps that's because they'll experience a large environmental risk. Or perhaps they have other genetic factors that aren't currently captured by these genetic scores. So again, this is to kind of make the point that genes are and genetic influences aren't independent of the environment. It's always within the context of the environment. So even in these situations, the environment always matters. And where I think these genetic scores are most useful currently is actually for research. And one of my areas of research at Birkbeck is focused on how to use these genetic scores to better understand how differences in DNA relate to differences between us. So I think about this at the level of the brain. Do these genetic scores or do differences in genetic scores between people alter how we attend to social information? Does it change brain processes? I'm also interested in about how these genetic effects unfold across development. How early do they come online and do they change across our life course? And again, it's worth me pointing out that these links between DNA variants and outcome, there's a lot of stuff going on in between there. DNA is in the level of the cell and we're measuring complex outcomes typically in adults. So I'm doing a lot of work at the baby lab at Birkbeck thinking about how these genetic scores impact infant brain development and how that might change. And then finally, again, I'd like to think about context. It's nature and nurture. It's not just nature. It's not just genetics. We know genetic differences are important, but they're not the whole story. And I really think that these genetic scores will help us better understand the role of the environment and specifically how they might influence how we move through the world and interact with our environment as we develop. So I think this is my, my last slide. So I hope in this talk I've shed some light into the answer to this question for you, or perhaps simply inspired you to go and look up behaviour genetic research, or think about a life in a STEM subject. But the bottom line is that DNA differences do matter, but they're only part of the story. Any individual genetic variant has an impact, but it's teeny tiny, and there are lots of them. We've got a much better understanding of which DNA variants matter, but there's still a lot of work to think about how. And finishing now, I've probably gone way over time. I'm sorry, Julie. Thanks. Obviously, the research I've talked about isn't just from me, it's from me and my colleagues and collaborators at Birkbeck and my gel lab. And obviously, thanks to my funders and research participants. Thanks, Julie.
Thank you so much, Emma. That was absolutely fascinating, particularly, I mean, for me, who doesn't have a background in genetics, just trying to understand some of the basic concepts and the terminology. So I'm going to have to ask you a question or two just to clarify something. So I've got my little spirit scientist doll here that my daughter made of me. So this is me.
Oh, wow.
Scientist's mum. You can see she's got beautiful blue hair and greenish blue skin. So Emma, would I be right in my understanding of the genetics here that if we wanted to understand the difference between a physical characteristic like hair color, so my brown hair or my brown eyes and her kind of yellow eyes and green hair, that these would be simpler genetic factors to understand and how they are different between humans versus if I wanted to understand my reading ability to her reading ability, maybe that's more complex because it's not such an easy thing to understand what it is.
Yeah, exactly that. When we're thinking about, I think, so eye color, for example, is often used as an example of a single gene kind of characteristic. And I don't think it's quite as simple as that. So it's somewhere between a kind of single gene and these highly kind of polygenic, lots of genes and the environment characteristics. But yeah, there's always this nice range. But certainly when we're thinking about differences in how we think and feel.
Okay, I've got
a message, lots of genes.
Yeah, that makes sense. And that was really clear from your talk. So that was really interesting for me to understand better too. So I have another question for you. And this might, you got to tell me if we need to myth bust here or not. Because I've always heard in the media, and I study gorillas and chimpanzees and other great apes who we're always told are very genetically close to us. We share 98.6% of our genetic traits with chimpanzees. But then I've also heard that we share 70% of our DNA with a banana. So is that true? And then what does it mean? Like, how close genetically do you need to be to another species in order for that to be interesting?
I don't think I know the answer to that. Also, it's
70% with a banana.
Yeah, I mean, it's true that that there's a really nice statistic about how I think when we're looking at. So I think what we need to think about in this position is we're looking at the average. So you're taking all humans and average and compare that average to banana average genome, and it's very similar, but that's because our cells have to do certain things to function. They need to be able to produce energy, and so those are the things that are similar between a banana and a human. And then again, with those differences between us, that's still a lot of DNA sequence. But that's a true fact. We are very close as human species to a banana genetically.
That's so weird, isn't it? But because you need to understand the scale, don't you, to understand what that means. Cool. So we do have a question from Alexandra asking, are Bayesian statistics used in research other than frequentist statistics?
Yes. In fact.
And you might explain what that means.
I did not plant this question, I promise. I am reading this book. Shout out for this book. But yes, Bayesian statistics. So I have just mentioned this kind of comparison of looking at differences in allele frequencies between cases and controls. Typically, we perform a frequentist type of statistical test using linear regression or logistic regression, but there, particularly in genomics, there's a move towards more Bayesian approaches and using the information in a better way. So the answer is yes. Cool. And I'm learning it.
And we're going to take one more question, and then we've got a few coming in. So we'll save those for the panel at the end. So do keep asking your questions in the chat for Emma, and then you can ask your questions for Katherine coming shortly. Katherine, do join us here, and we'll get going on your talk in just a second. But one more for Emma here before we introduce Katherine's talk. We've got a question from Alberto saying, if we know genetic differences impact the way we think and behave, would it be fair to apply law depending on each person's genome? I know this is not a science topic, but is it somehow linked?
Well, it's a question that should be asked. The answer is, I think, no, because these DNA differences, whilst they're explaining differences between us, they're not the whole story, and they're not perfect. And even if we had a much clearer understanding of all the genetic differences that relate to differences between us, it's still not the whole story. And DNA differences are always context dependent. We live in an environment, right? It's you can't separate the two. So the answer is no, I think. But there is a lot of work thinking about, and I think we all have to think about this as scientists and policymakers and the general public, thinking about if these genetic scores do get better, how would they be used in a fair way and in a way that's not going to increase inequality between people?
Fantastic. Very cool. Emma, don't go away, but because we're going to come back for a panel. Catherine, we're really interested to hear about your talk now. You're going to tell us about how the environment influences your physical well-being. So if you're ready to kick off and share your slides, I'm also going to mute my video and audio and listen to your talk.
Okay, thank you very much, Julie. Hopefully I am sharing my slides, my screen now. No, it's frozen.
Yep, we're all good.
We're all good.
There we go. Yep.
So yes, so the title of my talk was coughing up a lung, pollutants in our air, and this is my really bad graphic here, and I'm trying to show this poor unfortunate person coughing up an entire lung. And hopefully when you talk about coughing up a lung, most people don't actually imagine losing their entire lung, but in fact it's, you cough up a lung and you get that phlegm, and if you're out in the countryside, maybe it looks a bit phlegmy, and that's fine. And sometimes in the city, you look at it and you think, did I roll in the gutter? That looks pretty awful. What actually is going on here? And that's really what I wanted to just discuss in my talk. So we quite often say, go outside, get some fresh air. It'll improve your health. So what is fresh air? I've sort of pointed it in the sky here. And you think, well, what's in? This is the fresh air. So this is the atmosphere that we breathe. And obviously the atmosphere is mainly nitrogen and oxygen, but we'll all, I think, be aware of other species that would be present in the air, other chemicals that might be in the air. So for instance, obviously there'll be some CO2 in the air because we're all breathing. But there's actually thousands and thousands of other things in the air at what I'd say trace levels, just really, really low levels. And these might come from natural emissions, from plants, from animals, and they also come from unnatural emissions, and that's from us. So I'll talk about probably sort of the one most people would think about for the pollution sources anyway, and that's exhaust gases. So I've got my two pictures here. I don't know if you can see my pointer, I hope so. We've got the internal combustion engine. So the internal combustion engine, this is really what you have in cars when you're burning fossil fuels, when you're burning petrol or diesel. And the actual exhaust gases that you get depend on what sort of car you have. So here we've got a petrol car. And in its exhaust gases, it's got some water, but it's also going to have some CO. So this is where the car engine hasn't quite burnt all the petrol completely properly. You've got incomplete combustion, and you've got some CO. You've got hydrocarbons. This is really unburnt fuel. And then you'll have some what I'd call NOx. These are nitric oxide, NO, and nitrogen dioxide, NO2. And these can all be coming out of your car exhaust. Now, you might not have a petrol car. You might have a diesel car instead. This obviously is a particularly poor example of a diesel engine here, emitting some really, really very, very extreme pollution from its exhaust. But in diesel cars, the fuel is burnt a bit differently. Diesel's not quite the same as petrol. You burn it under different conditions, and the actual composition of the exhaust gas is a bit different. You don't tend to get so much of the CO, but you will have some hydrocarbons, some unburnt fuel coming out, and you will get particles. Obviously, there are extreme levels of particles in this exhaust here, but even in a much better behaved diesel engine, you'll get these tiny particles. And it's the tiny particles really that we have to be careful about. So we think about the pollution coming out of car exhaust, but actually when we're talking about what's in the atmosphere, it's actually nature. It's actually plants give us the huge amount of organic compounds, of carbon-based compounds in the atmosphere. So these are all the green plants. So I went out and took some pictures the other day. So there's this molecule here. I would call it isoprene, but if you're doing your chemistry at the moment, you might call it 2-methyl-butadiene, but I would always call it isoprene, and that's going to be emitted from green plants, along with what I'd call the terpenes. So these terpene molecules, they're hydrocarbons. They've got carbon and hydrogen, and that's all in them, and they're unsaturated. They've got double bonds, and we're all aware of these because we can smell them. So we've got the alpha and the beta pinene, and conifers, fir trees, these release these pinenes, but also all trees will release their own unique combination of these molecules, and that's what gives them their lovely scent. So you've got here some apple blossom, and that will have a particular combination of emissions coming from it to give it a lovely scent. Shown here limonene. So no prizes maybe for this one from guessing if you've got citrus trees. If you're in California, you've got the wonderful orange trees or the lemon trees there, that they will be emitting limonene. There's also, though, some more unusual ones. So things like methyl salicylate. Over here, I don't know if anyone knows this molecule, cis-hex-3-enol. So it's an enol because it's got an alcohol, and it's a hex because it's got 6 carbons, and it's an ene because it's got a double bond, and it's a cis because the double bond has got the same geometry on both sides. And it looks fairly complicated as a molecule, but what that is if you've ever been outside on a summer's day and someone's just cut the lawn, and you get that really distinctive smell of fresh mown grass, that is a bottle of cis-hex-3-enol. It's exactly the same smell. If you open a bottle of this, it just smells of a fresh cut lawn, takes you back to those summer days. And it's not just the green plants on the solid earth, growing in the soil. If you look out over the oceans and over the seas, then if you've got, say, seaweed there, you've got algae, they'll be emitting also up into the atmosphere. A different range of compounds, so they have their specialities. So here I've shown dimethyl sulfide, for instance, as a molecule that's being emitted up into the atmosphere by the plant. So nature just emits huge amounts of these organic molecules into the atmosphere, and I would call them volatile organic compounds, VOCs. And by volatile, it just means that they've got a presence in the atmosphere. They're not completely in the liquid phase, they've got some presence in the atmosphere. So for instance, with the terpenes, we can smell them, and they smell quite nice. Now, the trouble is that we'll all have heard of this concept of, or a lot of us will have heard of this concept called photochemical smog. And this is this pollution event that you get on sunny days when the air is polluted, and it's got a very distinctive characterization. So let's look at the key players involved in this. The first one, nitrogen dioxide. Now I've colored it this sort of orangey brown color because nitrogen dioxide is an orangey brown gas. And you will get some of this emitted from car exhaust, and it's certainly harmful to us. But the chemistry in the atmosphere is actually quite complicated. So you've got nitrogen dioxide, it's emitted from a car exhaust. Then on those lovely sunny days, the sunlight will break down the nitrogen dioxide. And it breaks down nitrogen dioxide, and you get this molecule called ozone being formed. Ozone's unusual. So regular molecular oxygen that you breathe is O2, and ozone it's very similar, but it's O3. It's got an extra oxygen atom. So it's got an unusual structure. And it's certainly a sneaky player in the atmosphere. So some of you might have heard, I don't know if anyone wants to put in the chat, but it's definitely got a good role in the atmosphere. It's got a good role, but it's also got a bad role. So if you look right up high in the atmosphere, so in where I'd call the stratosphere, way, way up high, far away from the air we breathe, there's the ozone layer. And the ozone layer is really, really important, because what happens is the ozone absorbs the incoming short wavelength UV radiation and stops it reaching us. It stops us damaging our DNA. It stops us damaging the plant life. It's really important to take out this really harmful UV radiation. So we need our ozone in this layer in the stratosphere. But down ground level in the air we breathe, we don't want it because it's harmful to us. And it's a secondary air pollutant. That means there are very few sources that are actually directly emitting ozone into the atmosphere, but mainly it's coming because we emit the NO2 into the atmosphere, and then the ozone is being formed in the atmosphere by the chemistry occurring there. So in our photochemical smog, we've got nitrogen dioxide, we've got the ozone formed by the nitrogen dioxide, but then we've got this other key player. These are these volatile organic compounds. Now these could be coming from, say, your car exhaust as unburnt hydrocarbons, but there's the plant emissions are the dominant source of VOCs in the atmosphere. The trouble is, if you've got VOCs in the presence of ozone on a sunny day, you lead to the formation of more NO2. What happens is the NO2 breaks down, it gives you NO, and then the reaction of ozone with VOCs converts the NO back into NO2 again, which means each time you go around the cycle, you make some ozone and you reform your NO2, and it's still sunny, and your NO2 breaks down, it makes some ozone, you reform your NO2, it's still sunny, it breaks down, you make some ozone, you reform your NO2. So on these sunny days, you get trapped in this sort of merry-go-round cycle where you keep making more and more ozone. And that is the problem. And then the very fourth thing that's important for photochemical smog are the tiny particles. Now, these are the ones which give it its characteristic appearance. The The particles of concern aren't the ones that you can see. So if you're coughing up that lung, cough, 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 and you're looking at your tissue and you've got all this dirty looking phlegm sitting there, that's great. That means your phlegm has done the job that it's meant to do and it's taken out all of these big particles. But they are not the ones of main concern. The ones of main concern are the really tiny particles. So if I could grab one of my hairs, and you can do this yourself, and look at it end on, your hair is about 0.08 millimeters in diameter. And you can just see it. You can just sort of see the cross section of your hair if you look at it end on. Now, if you imagine putting 100 particles into that cross section of hair, those are the sort of tiny sized particles which start causing the trouble, that size and smaller. So we're talking about really, really tiny particles. We can't see them, but they're present in photochemical smog, and they're present from two sources. They're present from car exhaust, and these are particularly diesel cars have a tendency to do this, of emitting these tiny particles. But they're also present from the breakdown of these VOCs in the atmosphere. Because what happens when VOCs break down in the atmosphere, these volatile organic compounds actually become less volatile, and they start forming these little droplets. So what you have are all these thousands and thousands of tiny particles. You can't see them, but they scatter the light. So when you look, the light is being scattered out of your field of view, and it gives the air a very hazy appearance. And it means you can't see, for instance, as far into the distance as you could do on an unpolluted day. So just to sum up, your photochemical smog, you get it on sunny days. You've got NO2, which gives us an orangey appearance. You've got these volatile organic compounds, could come from cars and in human activities, but also very much from plants. You form ozone, which is very harmful, and you have all of these tiny particles. And look here, this is a photograph. So this is a photograph I took the first day after the lockdown was lifted in April. So this is April 2001. The lockdown had been lifted the day before, so this is the first day we were told we could leave our houses. So I'm walking across Waterloo Bridge. The message was still stay home if you can. So there wasn't much traffic on the road. This was actually a really low traffic day, but beautiful blue skies. It was sunny. And if you look here, this is looking into the city of London. You can see, I haven't altered this photograph in any way. You can see this hazy appearance. You can see this orange look. And I didn't walk into central London, but I'm sure it was a bit smelly. So this is the photochemical smog, the nitrogen oxides, the ozone, and this hazy appearance. And what I'm interested in my work, or part of it, is how does this pollution, how does this photochemical smog interact with your lungs? Now, there are lots and lots of different studies that you could do here, and different scientists have their different specialities. What I'm interested in is a very particular bit of it, of what the pollutant does when it hits the very surface of your lung. So here's a pair of lungs, and this is your I'd call it a windpipe, you could call it your trachea, and the air is going to come down the inside of this pipe. And it branches off here into bronchi and bronchioles. And at the very end of these, you've got what are called your alveoli. People say these look like bunches of grapes. This is really important that you've got these tiny little sacs. They've got an opening of about 0.2 of a millimeter across to give you lots and lots of tiny little sacs. And you need these because you have millions of these tiny little sacs, and it gives you this massive surface area on the inside of your lungs. So the actual surface area of your lungs is about, I think it's about half a tennis court in size. And you need that because your lungs are by far the dominant mechanism of getting oxygen into you. So all the oxygen has to go across the surface of your lungs, and all the CO2 comes out through the surface of your lungs, or pretty much all of it. So you need to maximize the surface area. And you do that by having all of these millions of alveoli. But your bodies are a water-based system, we're wet systems. If these alveoli were lined with just pure water, you would have a problem because water has what is called a high surface tension. Essentially, the molecules of water have a stronger interaction with themselves than they do with air. So they try and minimize their exposure to the air, which means if you had these alveoli coated with just water, one, it would take you a lot of energy to expand and compress your lungs. But two, when you expand your lungs, you're okay. When you start compressing your lungs and you breathe out, they would just seal up. And obviously the reason you have these alveoli is to give you a large surface area for your lungs. And if they all start sealing up, you get a small surface area for your lungs. And that gives you obviously breathing problems. So to get around this, we have a layer of material called surfactant material spread at the very interface of your lungs. So the atmospheric air that you breathe in is here, and then this is the liquid on coating your lung cells, and then you have this film just spread out over the surface. So this fluid, it's called a lung surfactant, so it's like soap. So soap's a type of surfactant. So it's just a soapy fluid that you have over your lungs. It actually develops quite late during pregnancy. So if you have a very, very premature baby, then they can lack lung surfactant and they will have breathing difficulties at birth. This is why we know it's so important that you have healthy lung surfactant, because we can see the problems that the premature babies have if they're born very early without it. The surfactant itself, it's not just the detergent you could buy off the shelf at the supermarket. It's actually a really complicated mix. It took a long time to find out what was really required in it, but it turns out it's lots of different species play a role in lung surfactant. So you have what are called lipid molecules, and these give you a nice low surface tension. But on their own, it's not enough. You need some saturated lipid molecules, some unsaturated lipid molecules, some different types. You need some proteins. There's a particular protein, surfactant protein B, which is very important in maintaining this layer. And you need a species called cholesterol. And again, cholesterol gets, I think, a bit of a bad press. People talk about it in quite negative terms, but you absolutely need your cholesterol in your lipid layers and in your surfactant layers. So it's vital that you have this correct mixture of lung surfactant. And what we've been looking at in Birkbeck is just how air pollution actually interacts with this mixture of lung surfactant. So what we've got is we've got our lung surfactant spread at the surface. I'm just trying to show all the different colors. I'm trying to show you it's a mixture of different types of lipids. This yellow blob here is my attempt to draw on cholesterol. And this is my attempt to cartoon it a protein. And the question is, instead of the fresh air reaching the lung surfactant, what happens when we take this photochemical smog air to reach the lung surfactant? What happens to that? So for what I want to look at, we're looking at just the interface. We're looking at literally just that single molecule layer of lung surfactant spread on the surface of the fluid. So it's not a bulk technique. We're not looking at the sort of a beaker full of reagent. We're looking at just an interface of reagent. So we need techniques that we can look at just the surface. So to look at the surface, a good way of looking at a surface is to look at reflection from it. So we have different techniques that we'd use to look at reflections from surfaces. We might be able to use visible light. So for instance, we have used a technique called Bruce triangle microscopy, where you can look at the organization of material at a surface. But if we look at this layer here, it's a single molecule thick. So it's about maybe 0.2 of a nanometer. So it's 0.2 of a billionth of a meter thick. It's really quite thin. Whereas visible light is about 400 to 600, 650 nanometers in length. So the wavelength of visible light is much longer than the thickness of the layer that we're trying to probe. And that limits it somewhat, the information we can get. So what we would do is we try and use shorter wavelengths than visible light to look at the reflection of wavelengths which is shorter than that of visible light. But it's the same principle. We have light coming in and it bounces off the surface and comes away. So it's like a gymnast comes in, hits the mat, comes away again. And really, by looking at the reflected beam, we can say something about the mat that it hit. So if it's a nice, plush, deep, comfortable mat, or if it's a very thin, uncomfortable mat that the light bounced off. To do this, we need these short wavelengths. So actually, we quite often would use X-rays for this. So X-rays with a wavelength of say 0.1 nanometers, so really short wavelength light. We can't do that at Birkbeck. So to do that, we'd go somewhere else. So for instance, we'd go to, this is Diamond Light Source out in Oxfordshire with this big ring structure here with instruments off it. And we can go there and do our experiments. Alternatively though, we can get some different information. If instead of using X-rays, now X-rays, it's electromagnetic radiation, it's photons of light. Something a bit different which has a wave is a neutron, and you've probably come across neutrons as part of the nuclei of atoms. You've got protons and you've got neutrons. But you can also have neutrons acting as waves, and we can bounce those off the surface. So to do that, we can walk sort of 10 meters across that fresh air of Oxfordshire from diamond light source across to ICIS, pulsed neutron and muon source, and use their neutron facility. So the neutrons here would be going, this is the neutrons down here. They'd be fired out along here, and then they come off the instruments along the side of this main beam of neutrons. And just to say that science isn't glamorous, you can see these little porta cabins along here. These are where the scientists do their work. And I don't get to go in one of these porta cabins normally. I get to go in this one under here. So it's a porta cabin under a porta cabin, which I think is a truly glamorous place. But it's a fantastic place to work, actually, ICIS, because there's such amazing equipment and such amazing people supporting the work that you do there. And obviously you can see from this photograph just what a busy place it is in terms of the amount of equipment set up. And I don't have to go to ICIS in Oxfordshire. We can also go to France, go to Grenoble. In Grenoble, they make their neutrons in a completely different way than they do in ICIS. So here, this is, I think it looks like it's described to me as a top hat frequently. It's an aluminium shell, and this is a nuclear reactor. And that nuclear reactor produces neutrons that's used for science, for all areas of science. So if you go there, you can talk to scientists and engineers and technical people from huge range of different backgrounds. So you can talk to everyone, find out what's going on. I go there to look at lung surfactant, but people go there for all kinds of drug treatments and for batteries and for space studies, everything really. And also you can see from this picture, it's also just near the Alps. So if you did ever manage to arrange your time better, you can go out and have walks in the beautiful Alps or go skiing in the winter or whatever you can manage to do. Just to show you an experiment, obviously you want to actually do an experiment. So here we're looking at some lung surfactant, and this is a student, and she's just putting some lung surfactant down on this sample holder here. And this particular one was done at a diamond. So this is the X-ray source would come out here, and the X-rays just bounce off the surface and go up here into this detector. And then we can see here, I won't go through the data, but you can see if we had the reflection from just water, it's this gray line. If we had the reflection from our lung surfactant, it's this black line. And then if we expose it to just a really low level of ozone, as you'd find in photochemical smog, you'll see that it's changed. And this red line is quite different to the black line. And we've actually found out in this experiment that we've thinned our surfactant layer. It's become a bit thinner. So overall, if we do these experiments with our neutrons or with our X-rays and then back in the lab, or this is a booster angle microscopy image here, what we can see is when we expose our lung surfactant to pollutants in photochemical smog, actually a lot of changes occur. So in this image here, I'll say it's lipid gymnastics. Here's our healthy lipid at the start. The ozone comes along and it starts attacking the legs of the lipid, bits and fly off. So this might fly off into the atmosphere, or worse still, it might go and into the fluid beneath your lung surfactant and cause inflammation that way. And what remains, your sort of frail lipids which remain, they can start rearranging themselves and bending over. Or here, this is showing just the organization of your surfactant on the surface. You've got these beautiful round domains here of the normal surface. And then when we expose them, this is exposed to those tiny particles. So there's nanometer particles of sort of carbon material you might get from an exhaust. You can see they form these sort of angry looking shapes here. You can't see the particles, you're just seeing the effect that the particles are having on the surfactant. So that's everything I wanted to say, apart from the fact that there were lots of people involved in these experiments. So there were lots of students involved, and there's lots of other scientists involved in it. And I'd also like to add that if you go and do these experiments, it's not just doing science, there's also all the engineering and technical aspects. So for instance, if you go to ICIS or Diamond or in Birkbeck, there are people involved in actually building instruments as well. So if that's more your interest, there's plenty of opportunities there. And thank you very much for listening.
Thank you so much, Katherine. That was really interesting. And so interesting to understand that we can make organic, natural compounds that are produced by plants and should be, not dangerous. We are actually making them dangerous by the combination of them with our diesel exhausts, and how important it is that we phase those out. And hopefully that's the direction we're going. I wonder though, if you could tell us, do you think with COVID and many people wearing medical grade masks, will this have had any impact on the health of our lungs while we're in busier, more polluted areas?
So I think it's a really interesting question, Gilly, and lots of people I think are starting to look at this. So I'd say the first thing is that I don't think it's known yet, but there's certainly a suggestion that people who have lived in more polluted areas were more susceptible to becoming ill with COVID. So there has been a lot of talk, and we don't know the answer. It'll take a while to find out, but there's that suggestion. With the medical grade masks, there is the hope, I guess, that they might at least help filter out some of the particles before they get to the lungs. But do we really want to wear masks forever, or would you rather just not have the particles there to start off with?
Oh, sure.
Yeah, but definitely that is an area of active interest. It's like how much did it help improve when you're wearing the mask to not have the particles go in the first place. And you're completely right as well about the vegetation in the trees. That's what's there natural. We need to make sure that whatever we do doesn't cause an adverse effect from that, I think.
Yeah, and I'm sure it took us a while to understand how that unfolds and how we were damaging the planet based on those interactions. Well, damaging ourselves with those interactions as well. Yeah. Oh, so we've had a question come in from Lisa asking, first saying, thanks so much for your talk. We've had a few saying, thanks so much for your talk. Can I ask, is ozone more damaging than PM 2.5? And you're going to have to tell us what that is.
So that's an excellent question. So PM 2.5, these are really the tiny particles that I was talking about. So these are particles with an aerodynamic diameter less than 2.5 micrometers. So the nanoparticles would fall under that category. So when you've got big particles, they don't tend to get into those alveoli, into the very bottom of your lungs. They tend to get trapped on the mucus, and you cough them out and you see them on your tissue and you think, yuck. But that's an end of it. Whereas with the tiny particles, they get right deep into your lungs and they can embed into your lungs. And the ozone can also get right deep into your lungs and then react with the surface of your lungs. That's the problem. Which are more dangerous? I don't know if I could say both are dangerous, I think is the answer. It depends slightly, perhaps, on what Emma was saying on genetics, how susceptible people are to different problems, because for one problem you might get an inflammation response, say, from the ozone and the byproducts of that. The particles, I think there's more unknown still about the particles and what they're doing, but we do know they get taken up into the body and you can see them sort of distributed around the body. It's quite scary, but when you look at the hospital emissions, of course, if you've got the particles, you've also got the ozone on those days. They're always linked together, so you're always getting both in the air. It's hard to say, sorry.
No, it's a great question. Thanks, Lisa, for that one. I'm gonna ask Katherine to stop sharing her screen, and I'm gonna invite Emma back into the panel. And a couple of the things that we talked about early on before we started the talks were, can we find out a little bit about how each of you got into your field? So we know that obviously you've had to do quite a lot of studying. Did it make a difference which A levels you took? And did you know what you wanted to do immediately? And did you have to know what you wanted to do? Can we find our way into these jobs later? Do we have different paths? All that sort of stuff we're interested to know about. So Emma, let me put that to you first.
Thanks. And it's a great question, isn't it? And I think we have this view that scientists are kind of mini scientists from primary school and secondary school, and this is what you're doing. So for me, I didn't know really what I liked, other than I was interested in science and biology generally, and I did psychology as an A-level, really enjoyed it, but then switched back to biology for my undergraduate, and it was in my undergraduate degree that I had a module on genetics. It was only, I think, an 8-week module, but that was the moment of me thinking, this is it. This is linking psychology and biology. And even after that, it wasn't quite clear cut where I would go. So I completed my undergraduate degree, still didn't really know what I wanted to do. Other than I knew I didn't want to go and just work in an office. I wanted to go and have some practical experience, work in the lab a little bit. And then I went on to do a more specialised master's course that I selected because it had wet-based lab research component. And then I pestered people after that. It's like, can I come and join your lab? And thankfully it worked. And I ended up doing my PhD under the supervision of Professor Robert Plomin over at King's College London. And yeah, I fell into behaviour genetics research really, but it was more of a matter of I kept following what I liked and what seemed to suit me and drilled down from there. But I would never have thought this was what I would end up doing.
But I think your story in your pathway demonstrates the fact too that the subject areas that you study in school, the boundaries between those subject areas are not fixed. And actually you can be in psychology and studying biology, and you can be in biology and studying psychology. So like you don't have to think as an A-level student or a GCS student like, oh my gosh, I'm now stuck on this track and biology is it for life. Because actually what we're finding that the more methods that we're using and the different ways that we're studying who we are and how we came to be actually involves many different disciplines. And I think that's quite exciting. Katherine, what's your story?
Well, I suppose I had no idea what I wanted to do at school. I honestly, I feel like I drifted through that part. I went to school in Scotland, so I did higher rather than A-levels, which had the advantage you do more. So I didn't have to specialise until a little bit later. But even in university, I would say it was only in my final year where I started doing research that I thought, oh, this is what I want to do. This is fun. I like the freedom of being able to explore different questions. And yeah, that's drifted on from there, like Emma, just doing what I enjoy doing, I suppose.
Cool. And what about this idea that science is very rigorous? We're always just, in labs, it's quite strict. You have to be very disciplined. It might drive a lot of people who are more creative, who like the arts, who want to be able to develop new ideas and use their imagination. It might kind of deter them from entering the sciences. Emma, I'll put it back to you. Do you think there's any creativity in the work that you do?
Absolutely. And actually, if I'm honest, I think the best scientists I know, I think that's the key. What I notice about them is they are very creative. And even thinking about routes into science, again, some of the best scientists I know in terms of the novelty of their work and the insights and how they go about their research is informed by their background, which is not necessarily science. I've had, I know great scientists who come from philosophy, who've come from a background in music.
Yeah, philosophers are great.
Yeah, no, exactly. And I think it really helps because it informs, you think differently. And that's really what science needs, is people who have had a diversity of training and they think in different ways, because we might be interested in this one question and we have to apply certain rigorous methodology and statistical procedures, but there is no one way to answer that question. It's, you need to come at it from different angles, and each of those methods will have its own limitations. So yeah, and sometimes it's a creative thought of thinking of a method that exists in a completely different discipline and saying, hey, let's take that, and we might try to use that to look at something in biology or psychology, and that can kind of change everything for that field. Yeah, what about you, Katherine?
Well, I mean, if you're doing research, obviously you're doing research that nobody has done before and nobody has been able to do before. So you have to think of something different, a new approach, a new question, a new angle, which is why I love talking to people in other fields, because you learn so much from just talking to, I go to places and you talk to archaeologists and they're doing this and that, and you find out what they're doing, or you're talking to somebody who works on batteries or mechanics or something, aerospace or something, but it's just talking to a huge range of people across the arts and the scientists, and you get a much better understanding of the questions that you can ask or should ask.
And I think you raise another interesting point there, Katherine, too, about being a scientist doesn't necessarily mean that you're doing the same thing every day. You're probably doing a wide range of things, from teaching and public engagement like this, and working in the laboratory. But you're also getting to travel and share your research around the world, right? Obviously, in COVID, we're doing that on Zoom, but you know, in normal pre-COVID times, and hopefully very soon again, you might be traveling to different countries where you're going to be sharing your research with other experts in the field and sharing your thoughts and ideas on it, which is thoroughly exciting opportunities there. Okay, we got a couple more questions in our chat stream, if you're happy to take those before we wrap up, because we've got a few minutes remaining. We have another question from Lisa to Katherine about those tiny little particles. Do you know if the PM 2.5 captures organic matter too, or if particles this small are only the pollutants that we are generating?
No, so the organic matter, well, when we have oxidized the biogenic particles, whether you call them natural or not is I suppose, an unusual question, because we have helped in how they have degraded depends on their interaction with us. So they might degrade one way in a clean atmosphere and a slightly different way in a dirtier atmosphere. But the actual definition, yes, those particles are tiny, so they come under the 2.5 micrometer and less range. With science, always as the caveat, it depends slightly on how you're capturing your particles and whether you're going to detect them or not, the actual detection method that you use, but they do count in that size range, yes.
Oh, and we've got a couple more for you, Katherine, if that's okay. Lisa also has asked, you mentioned that there are smelly elements to smog, but you didn't mention what's making those odors.
Oh, so ozone smells quite distinctively. There's a very distinctive smell you can get from ozone, and NO2 itself smells. But then you get more complicated molecules. So there's a molecule called PAM, which is formed from the breakdown of these organic molecules with nitrogen dioxide in the presence of oxygen, and it also has a distinctive smell. I would say not a very pleasant one. So it's that breakdown basically of organic molecules in the presence of the nitrogen oxide, in the presence of sunlight. You get this mixture, and it is a smelly mixture, but there's actually quite a few things in.
Oh no! Oh, we just lost that last 2 seconds of your answer, Katherine.
Oh, so for the smells, I guess the ozone, yeah, and the pan are probably the main ones, but there's a lot of things in there which smell.
Oh, and we'll take a final question for Katherine here. If anybody has any more questions for Emma, pop them in. We've got 1 or 2 minutes left. Alberto's asked you, since we're making ozone, could we find a way to move it up to the layer where it benefits life instead of damages it? Great question.
Brilliant question. Absolutely brilliant question. I think this is where we need our imagination as scientists to answer that, because that is obviously the ideal. If we could put it back, fill that ozone hole back up again with everything we're making down here. But I don't know the answer to that, but I hope there is one, and it's something to look at.
Yeah, great questions. Thank you for everybody who's put questions in today. I think that is bringing us towards the end of the session. Before you all go, I want to thank our speakers, Dr. Emma Meaburn, Dr. Katherine Thompson, both from Birkbeck's School of Sciences. And you can find out more about them on our website. You can also rewatch this video, which will become static on Birkbeck's YouTube channel. And I want to remind you that we are here for 2 more Saturdays. Next Saturday, we have the amazing Dr. Simon Green, who's going to talk about the biology and culture of psychiatric disorders. And he's going to be followed by Dr. Ute Lersch, who's going to talk about your brain and resilience. So how do humans deal with resilience, and how can we improve it? And in the final week, on the 29th of May, we're going to be having our Becoming Human theme. And I'm going to tell you a little bit about the evolution of our divided brain. Why do we have 2 hemispheres? Why not 1? And Dr. Natasha Kirkham is also going to tell you about how babies develop in the middle of everything in her talk, Baby Meets World, which Emma is going to host for us in that final week. So we're super happy that you've been with us today. We hope that you will share Science Saturdays with your friends, maybe your local schools and communities. We're going to keep them up as an open resource, and just hope that you also find out a little bit more about Birkbeck. You can also check out our Twitter feeds, the Me Human Twitter feed and the Birkbeck School of Science and Maine Birkbeck Twitter feeds to find out more about our sciences. So I think that's it from us today. Thank you again to Emma and Katherine. We're going to sign off and we'll see you next week.
Thanks very much. Bye-bye.